Hello everyone, welcome to today's webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I'll be hosting today's session on discovering and developing new treatments for fungal infections. We're pleased to have developed this webinar in collaboration with the Center for Infectious Disease Research and Policy, CIDRAP. Revive is GARDP's education and outreach program and was officially launched in 2018. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All webinars are recorded and can be viewed after the live broadcast on our website, revive.gardp.org/webinars. As well as our webinar recordings, you can also read our series of articles known as Antimicrobial Viewpoints, where experts discuss various topics within the field. The most recent being a contribution from Martin van der Heijden from the WHO on strengthening regulation of antimicrobial use. We also have a resources section which includes the Antimicrobial Encyclopedia. This has definitions of various important terms, and some of these include videos where experts give a further explanation. These resources will be continually expanded with new terms and videos, so please keep an eye out for them over the summer. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window in your webinar control panel, as shown on this slide. If your question is specifically directed to one of our speakers, please include your name at the beginning of your question. We will also address the questions after the webinar, after the presentations, and do our best to respond to as many of them as possible. Our moderator today is Marnie L. Peterson. Marnie is the Outreach Coordinator of Antimicrobial Stewardship at CIDRAP. Welcome, Marnie. I now hand over to you to introduce today's speakers.
Thank you very much, Victor. I'm here to introduce our very first speaker today of this webinar. Our first speaker today is Maurizio Del Poeta. Dr. Del Poeta is a distinguished professor at the State University of New York at Stony Brook in the United States. He has a broad background in infectious diseases with specific clinical training and basic science experience in this field, as well as biochemistry. His research group have identified and characterized specific new inhibitors of the synthesis of the fungal sphingolipid glucosylceramide. His work also focuses on fungal pathogenesis and innate immunity, particularly in the understanding of fungal virulence and fungal host interactions using the human pathogen Cryptococcus neoformans. We're very much looking forward to his presentations, and I am now happy to welcome Dr. Maurizio Del Poeta to turn on his camera and begin his presentation now. Thank you very much for joining us. I'll hand it over to you.
Thank you very much. Can you hear me?
Yes.
Thank you very much for joining. Hello, everyone, and thank you for joining in. It is a pleasure to be here. Thank you, Marnie, for the invitation, and Victor. Okay. Here we go. Next slide. Here we go. I must disclose my financial interest. I have arrangement with Microfluidics Technology because I'm a co-founder and chief scientific officer of this company. I've been working also with Gentum, and I was a principal investigator and consultant on Novartis. Next slide. Yeah. As a laboratory focusing on mechanism of fungal pathogenicity, and in particular, I've been studying lipid pathway, sphingolipid instead of glycosides. And a second part of my lab also, a different part of my lab also work on host susceptibility to fungal infection. And in here, we are very focused on the types of by single malin synthase 1 and 2, and the one regulated by SK1, the sphingosine kinase 1, and the sphingosine 1 phosphate. We found that these two pathways are particularly important for innate immunity against Cryptococcus neoformans and also other fungal infection. And obviously, as Marnie also said, my recent focus is on antifungal drug development. And if I have a next slide, please. And for antifungal drug development, we have three main projects. One project is to study several drugs that we found to inhibit the synthesis of fungal glucosylceramide. And this is also the focus on the talk today, of the second part of my talk. And we also working in identify inhibitors of fungal ceramide synthase because of a new discovery that STER1 is essential for grow under physiological condition of Cryptococcus neoformans, and therefore, and also the fungi, and also a drug targeting fungal ceramide synthase 1 will be highly fungicidal. The downside of focusing on fungal ceramide synthase is also mammalia has actually 6 different ceramide synthases. So here we are really looking at something highly specific. And a third part of antifungal drug development is focusing on try to identify inhibitor of stereoglycosidase 1, which is one of a fungal specific gene involved in stereoglycoside synthesis and metabolism. Next slide. Yeah, as I mentioned, I will provide some example later on acyl hydrozone here. So I was asked also to provide an overview on vaccine and immunotherapy. About the vaccine, I would like to highlight this recent review article, which was published in 2021 by Stu Levy's group. The article has been written by Lorena Oliveira. It's a very comprehensive analysis of current vaccine that has been proposed for human fungal disease. As you know, there is no vaccine for human fungal disease in the clinic, but we are very close. If I can have the next slide, please. During the last few years, there have been a lot of work on proposing vaccine for fungi. But the main problem for vaccine for fungi is that fungi are normally immunocompromised microbe. And therefore, the vaccine needs to work under immunodeficiency. And most of the time, the immunity that gives protection is exactly the immunity that is lacking when the fungus is infecting. Therefore, you know, the vaccine needs to work in condition of immunodeficiency, particularly that immunodeficiency that is causing the fungus to proliferate. But nonetheless, there's been a lot of work, particularly on 5 different type of vaccine: live attenuated fungi, kill fungi, fungal extract, recombinant protein, and nucleic acid. And here, I briefly highlighted the infection that has been tested. It's been tested with Cryptococcus, Candida, Aspergillus, Paracoccidioides, Coccidioides, Sporotricasi, Histoplasma, Blastomyces, Tararomyces, and Emergomyces, which is a new dimorphic fungi, which is in emergency right now. For live attenuated vaccine, obviously they are very good. They produce a very strong and long-lasting immunogenicity. The manufacturing also is very straightforward. You take your favorite fungus, you delete your favorite gene. The favorite gene is not producing a virulent mutant, and therefore this can be used as a vaccine. The problem is that, again, under this condition, you may risk a sustained infection in condition of immunodeficiency. Kill fungus has been also studied. Obviously, a kill fungus does not elicit any infection. The main problem with the kill fungus is that the immunity is not as strong as using a live attenuated vaccine. There's been study with fungal extract. Obviously, there has been the advantage of using fungal extracts that may contain numerous multivalent antigen. The downside is that it is highly specific to that specific extract that you are using. but the study has been done in this area, particularly with glucan particle containing Cryptococcus alkaline extract. You can have a purified protein, peptide, carbohydrate, and lipid. And obviously, this will minimize also the potential side effect. But again, here you may stimulate only a certain type of immune response specific for PET antigen instead of just covering the entire immunity. And then there is the nucleic acid encoded delivery of antigens, such as a DNA RNA vaccine. This is very nice because also is a fast manufacturing process that produces a strong immune response. But again, there is a risk for eliciting perhaps an unintended immune reaction. However, among the many vaccines that has been studied, there are 3, if I can have the next slides, please, that has reached clinical trial in human. One is the FKS, which is a valley fever vaccine, which is a vaccine that has been tried in mice and also monkey, and they're extremely efficacious in protecting against the coccidioides on both animals. But then when they did the investigator did the clinical trial, it was not protecting in humans. There are 2 additional vaccines that has been done in clinical trial. One is a PEV7 from Pevion Biotech, which is against Candida albicans, against vulvovaginitis candidiasis, and has been protective. But the third, the NDV3, in my opinion, is the one that is closer, extremely close to the clinic, as I believe phase 3 clinical trial has been just concluded, about to be concluded. And this is for recurrent vulvovaginitis, and is one of the most promising vaccine. Is made out of the N-terminal of the protein ALS3, and is highly protective for recurrent vulvovaginitis. Next slide, please. About immunotherapy, I would like to highlight this review, which was written by Moulana Adime and young faculty at the University of Addis Ababa. And there is a very recent review which has been published on Frontier in Cellular and Infection Microbiology. Next slide, please. This review highlights 3 major immunotherapeutic approach that has been tried against the fungi. One is an immunotherapy based on monoclonal antibody. Second one is an immunotherapy based on cell transfusion. And the third is an immunotherapy based on cytokine treatment. The one based on monoclonal antibody has been tried against a series of fungi. I would like to highlight the Cryptococcus, particularly the one that has been tried by using the anti-GXN. There is also another antibody with Candida, micrograph, and many other additional antibody that has been tried with different pathogen, producing significant and important results in animal. The downside of using the monoclonal antibody is that it's highly specific, and the production has been considered very high in the past. Now I think the production is much lower. The good thing about using monoclonal antibodies is that they obviously is very specific and avoid toxicity because they are directed specifically to the pathogen epitopes. And also the possibility to use a monoclonal antibody together with an antifungal drug development is very good because it may reduce the toxicity and the incidence resistance of the antifungal drug. About the immunotherapy based on cell transfusion, there has been a lot of studies in vitro, in animals, but also in patients, particularly using granulocyte dendritic cells that has been stimulated with specific antigen and then transfused into a new patient. And this has been tried with Candida, Aspergillus, Cryptococcus, adoptive T cell transfer, and very recently also a CAR T cell therapy that has been tried for antifungal for Aspergillus fumigatus. And CAR T cell is a very new approach. There is very highly specific and very potent immunity. The problem with CAR-T is that you will have what is called a cytokine storm, and the patient is going to get worse before it's going to get better. So using CAR-T for antifungal, it needs to be done in a specific and highly protected setting. Immunotherapy based on cytokine, two types, the colony stimulating factor or pro-inflammatory cytokine. And both of them, they are being produced significant important results, although none of these type of immunotherapy is routinely done in the clinic. But particularly the one using interferon gamma, IL-12, IL-17, and IL-36, IL-18, IL-7, and TNF alpha has done some production, some significant data, significant results, particularly in clinical trial. Next slide, please. Next slide, please. It is nice to see, oh, one back. Can you go back, please? It is nice to see that NIH is also involved in promoting, oh man, I need to go back. Next, previous slides, please. It's a little delay. Yep. Next, here we go. Now. Next slide, please. Here we go. It is nice to see that previous slides, please. Previous slides, please. Here we go. It is nice to see that NIH is also willing to provide initiative for the establishment of center for fighting fungal infection. And this is an example for fighting mycosis, which during the next few months, we will have a few centers working together with the many investigators in the country and try to fight coccidioidomycosis. And these are focusing mainly on developing of a better immunity, providing a platform for the discovery of new vaccine, and providing also a platform for the discovery of new antifungal that not only is going after coccidioidomycosis, but perhaps also other against dimorphic fungi such as blastomyces and histoplasma. Next slide, please. I won't touch the screen. I won't touch anything anymore. Victor, I would like for you to run and touch the slides. So next slide, please.
All right, sure.
So about the new antifungal, there is this nice review that has been recently written by Ariana, and is open on the horizon, novel fungal treatment in development. Next slide, please. And the reason why I like this review is because it highlights not only the new drugs in development, but also the stage in which they are. We have a new antifungal development that are targeting the cell wall, the cell membrane, fungal pathway, and intracellular target or intracellular unknown pathway. And as you can see here, there's been drugs that are in phase 3 clinical trial, particularly the kinocandin derivative. But there are also some new drugs that are in preclinical trial, and some drugs that are at the beginning. I invite everybody to just read this review and go over the pros and cons of this antifungal. Our compound is still Hydrozone, and they are on the preclinical trial, and I will discuss this a little bit later. I would like to highlight also the calcineurin pathway, particularly the APX879. This is a very interesting compound that has been developed by chemical synthesis, by modifying the structure of a current compound that target calcineurin. And as you know, inhibitor of calcineurin are also immunodeficient or immunosuppressive. But the APX879, what Amplix were able to do is to dramatically decrease the immunosuppression of the drug, but retaining the antifungal activity. I will go to the next slide, please. So the seal hydrozone that we discovered many years ago went from a very broad spectrum of activity targeting Cryptococcus, Candida, Aspergillus, and also a series of Blastomites and dimorphic fungi. We have been working on this for the past 15 years. We have been going a long way. In this initial treatment, as you can see over here, this is an in vitro assay. We were active, our compound were active mainly against Cryptococcus and some dimorphic fungi. Next slide, please. But now with the new compound that we have, particularly P3G8 and P312, we are now active against a variety of fungi, not only Cryptococcus, but also against Candida, Candida auris, and Aspergillus fumigatus, suggesting that this compound are very highly, very broad spectrum of activity. Next slide, please. Here is the data that I'll show you with the effect on P3G8 in animal model. I will show you the Cryptococcus, Candida, and Aspergillus. And compared to a parent compound, D13, you can see that P3G8 is able to have a 100% survival in a model in which the treatment is started on the same day of the infection, the intranasal model. Next slide, please. In the second experiment, when the treatment is started at 5 days post-infection, also P3G8 is able to protect about 90%. Here, P3G8 is indicated at SBFAF1002, but it's the same drug. And as you can see that if you start at day 0, day 5, basically you have almost an identical survival. Now, compare this to fluconazole, you can see in the lower panel that our drug is able to almost sterilize the lung upon the treatment. These are endpoint CFU, meaning they are CFU that has been done at the experiment. Next slide, please. And the effect of the 5 compound against Candida and non-albicans is illustrated here. And now you can see that the 312 is very active in vitro, particularly against the series of Candida auris. And 222 or against the series of Candida auris, and whereas fluconazole, as you can see, is highly resistant. Next slide, please. This is the effect against the Cryptococcus using different doses. And as you can see, when we are using up to 40 milligram per kilogram per day, you have a very highly 100% survival. The survival decreases when the concentration of the drug is diminished. Next slide, please. The effect, previous slide, please. The effect on Candida is also significant, particularly when the drug is given intravenous. When the drug is given orally, the Candida model that we have is highly aggressive, and the compound is able to produce some effect, but not as a good effect when the drug is given intravenously. When the drug is given intravenously, the survival is 100% after 13 days of infection. Whereas when the drug is given orally, consider that this is an infection that is given intravenously, so it's very highly disseminated. But we have also an effect, though not as significant as when the drug is given intravenously. Next slide, please. The effect on Aspergillus is also significant. As you can see over here, the administration of Aspergillus, administration of a drug is dose dependent, and the effect is 70% survival, which is comparable to the voriconazole, which is about 50%. When the compound is given prior to the infection, and we started the treatment 5 days before the infection, it was interesting to see that the drug is able now to protect at 100%. So the drug can be used as a prevention. Consider that this model is also highly aggressive because the infection is given after immunosuppression with corticosteroids. That's what IS means, immunosuppression. And so the effect, interestingly here, the endpoint CFU suggests that the compound is eliminating fungus from the lung, as you can see from the CFU illustrated in D and B, particularly when high doses are used. And it was interesting to see that voriconazole does not do that. Next slide, please. So the effect on sporotricasi is also very interesting. This is a paper published in collaboration with the Brazilian collaborator, and you can see here that D13 is able to treat the sporotrichosis very nice compared to fluconazole, almost eliminating the infection in the skin and the elimination of inflammation. Next slide, please. Now, when D13 is used to treat few cats, and I will show you here Mimico on the top, Pythagoras and Bob. As you can see that the treatment with D13 together with itraconazole is able to clinical cure this cat from sporotrichosis. Now, this treatment was only approved when the cat was refractory to itraconazole, meaning we can only treat a cat that did not respond for itraconazole for at least 1 month of treatment. And the D13 was approved only as a combination therapy with itraconazole. The success that we had, but because of we are now going after cats that has been virgin, meaning not treated with any other drug, and as a primary therapy. But the results that we have with Mimico, and Mimico has been treated with 90 milligrams per kilogram per day for 3 months, together with itraconazole. Pythagoras has been treated with also 90 milligrams per day for 60 days, whereas Bob for 80 milligrams per kilogram for 30 days. Next slide, please. I believe we have 2 more cats to show you. And one is Ariana, and Ariana has a small lesion. You can see has been treated with D13 and itraconazole for 3 months, and obtain a clinical cure. And the next slide, please. And Pretigno has also been refractory to itraconazole, and you can see this is a serious lesion, and has been treated with itraconazole and D13, 80 milligrams per kilogram per day, and for 1 month, and is improved, dramatically improved the clinical condition. Next slide, please. So this treatment with D13 and itraconazole in combination with itraconazole, or perhaps D13 alone, could be a good alternative for sporotrichosis in animals. Next slide, please. Next slide, please. Okay, so in summary, I show you the progress that has been done with the vaccine. I think it's a remarkable progress, particularly for study in animals. NDV3 most likely will be the first vaccine that will be introduced for human setting for recurrent vulvovaginitis. Can you go back to the previous slides, please? And the immunotherapy are highly specific. They are still exploratory. I think the cost is still very high. They potentially have side effect. A new antifungal drug, there has been a significant investment. And also because of the news, I think that fungal infection are increasing worldwide. I think people are more aware about fungal infection. I think that we are more immunocompromised than before. People are now, they're using a lot of immunosuppressive drug for chronic diseases. Multiple sclerosis is an example in which if you're taking fingolimod, you may be at risk of developing cryptococcus. So because of this increased incidence of fungal infection, there is a more need of a new antifungal drug. And I think that we will see more antifungal coming up in the future. Don't forget climate change. Remember that most of the fungus that is affecting us do not affect, most of the fungus they affect because they grow at high temperature. Most of the fungus, they cannot grow at high temperature. If they adapt to a new temperature in the environment, they may become a pathogen to us. And so I would like to thank, next slide, please. I would like to thank my group. It's been just fantastic to work with these people. The work that I do with antifungal is in collaboration. Next slide, please. The work that I work with antifungal is in collaboration with the group in Brazil and the Stony Brook University. The animal work with the cat has been done with Isabelle Gramiao. The work that I do for antifungal here at Stony Brook is with Krupa, Caroline, Nivea, Sridevi, Samir, and Veronica mostly work on immunity against crypto, and Tyler is more on antifungal vaccine development. Anyhow, I thank you again for this opportunity, and I will be happy to take any question later on. Thank you.
Thank you so much, Dr. Del Poeta, for your presentation and some of your very interesting groundbreaking work in new antifungal discovery and development. We will be able to ask him, our listeners will be able to engage with him at the end of this presentation. And now I would like to invite our second speaker today, Tejas Bouklas, Dr. Tejas Bouklas. So Dr. Maurizio, you can go off camera, and I'd like to invite Dr. Tejas Bouklas to come on camera for her presentation. She's an assistant, I'll go ahead and introduce her as they're doing so. She's an assistant professor in the Department of Biological Sciences at the State University of New York College, that's Old Westbury in the United States. She investigates the persistence and resistance of fungal pathogens, such as Cryptococcus neoformans, Candida glabrata, and Candida auris. I'll bring my camera back on. Her research group aims to contribute to the antimicrobial pipeline in response to the global threat of drug-resistant infections. Her work has been published in renowned journals, and she has presented at several international forums. And she is an American Society of Microbiology undergraduate faculty research initiative fellow, and is committed to advancing infectious diseases research and training of the next generation of STEM scientists, which she's also going to talk to us about today, which is extremely important. Not only developing the pipeline of drugs, but developing the pipeline of scientists that we need to develop those new technologies. So welcome, Tejas, and you can go ahead and start your presentation.
Thank you, Marnie and Victor, as well as CIDRAP and GARD-P for this invitation. I'm going to switch gears a little bit and talk more, I guess, to you about the process of research and student sourcing the antifungal pipeline, which is severely lacking, and new antifungals let alone new antimicrobials, have not been on par with the increasing incidence of antimicrobial resistant strains that are out there. We also have an incredible workforce out there in terms of our students, and I'll talk a little bit today about the primarily teaching institution where I work, which is SUNY College at Old Westbury. And so we have no lack of students, and we are an institution that has quite a few women and underrepresented minorities, and that's the focus of my talk today. Okay, Victor, I may have to ask you to move next slide for me. Thank you. So I wanted to tell you a little bit about my institution. I didn't start here. This is SUNY Old Westbury at the bottom down here. Beautiful buildings you can see. To those of our in the audience who are from outside the US and outside New York maybe might appreciate some of these pictures. I started at Albert Einstein College of Medicine. That's where I got my PhD. I then did my postdoc at Stony Brook University. I got my first faculty position at Long Island University Post, which is this very gorgeous building. And then I am currently at SUNY Old Westbury, which is also a state institution in New York, just like Stony Brook. My institution follows the trend that we see in most colleges. Most of our students are female, but we're also an institution that is incredibly diverse in our student body. More than 67% of our students are underrepresented minorities. The majority of them receive some sort of financial aid. Most of that aid comes from the federal government. And so prioritizing research is not necessarily something that these students are even thinking about. They're focused in just kind of acquiring their undergraduate degree to begin with. Next slide, please. So when I made my move between the two teaching institutions, between LIU Post and SUNY Old Westbury, the part of the reason for that move was we've been seeing classrooms with students interested in PhD and MD and other graduate schools, but not really going for research opportunities, which they would need in order to get admitted or even be competitive for some of those schools. And so there weren't enough faculty members to students for those research opportunities. And so in collaboration with the faculty at Post, we published this article on some of our initial efforts in integrating investigative research into the classroom directly, and not so much in a research lab. And this was to create some foundational research experiences for science majors as well as pre-health students who also were interested in acquiring more research and authentic research experiences. The kind of foundational experiences we chose, you can see a picture here. That's a high school student, actually, a local high school student injecting Galeria, which is a worm, with a syringe. And so those foundational experiences were tailored to our students, our undergraduate and graduate skill sets that they were coming in with. We focused on some basic microbiology and microscopy techniques, some molecular biology, tissue culture, antimicrobial research, and simple models of infection like the worm, not so much rodent models, which may not have been as available or accessible in our institutions. Next slide, please. So the motivations, or the bird's eye view of why we decided to student source this from the perspective of a researcher, that is, instead of simply having your small research cohort group in your lab, kind of came from this Presidential Council of Advisors on Science and Technology report, PCAST. That report said that we needed in the United States another 1 million STEM graduates in the next decade, or we were not going to meet our economic demands. The big proposal for that was to replace standard lab courses with discovery-based research courses, that is, authentic research experiences. And sort of the first recommendation for this came from Howard Hughes in a course called CPhages. This is really an acronym. It stands for Science Education Alliance, Phage Hunters, Advancing Genomics and Evolutionary Science. Next slide, please. And so what CPhages was proposed initially as was simple digging in the soil to find new viruses, that is your bacteriophages, and then students would progress through microbiology techniques, then to some complex genome annotation and bioinformatic analysis. And much of the protocols for this were developed by University of Pittsburgh in collaboration with Howard Hughes. Next slide, please. Here's sort of the research flow that students did, and I'll talk to you about how we run this course in my institution. But all courses that run CPhages start with students isolating and going into the soil. I mean, soil which has like 10 to the 11 bacteria really in like a gram, has an incredible resource for potentially new antimicrobials. Students then acquired a lot of wet lab techniques, including purifying, plaque morphology readings, amplifying much of what they collected, electron microscopy of their phages, more molecular biology involving DNA extractions, and even characterizing them for that further on. And this was all done within 3 months. Or less. That is 1 semester of an undergraduate course with a set of 24 students or so with 1 section. So at any given time, you have 24 students all working towards that goal, and therefore you're kind of maximizing the amount of findings that you could potentially have. Next slide. And so where CPhages today is sort of in review is it has successfully, to some extent, done what that PCAS report asked for. CPhages graduates show better retention, much higher retention, much better grades compared to those students who take a traditional biology course in undergrad. And the course has been implemented by over 4,800 students. We're over 20 cohorts in now at 73 American institutions. And those institutions are not necessarily just teaching institutions, but also, as you can see, most of the institutions are research intensive universities, like some of the ones where I came from. At SUNY Old Westbury, it's taken by honors biology students in their first year. So you can imagine if they're not non-traditional, these are 18, 19-year-olds coming in with very little research experience, completing that entire research flow, and publishing themselves their phages and characterized phages that they acquire. So as a researcher, you're making a lot of progress incredibly fast as well. Next slide. So CPhages was very focused on bacteriophages. I'm not a virologist, right? We're here today to talk about antifungals and developing and enhancing an antifungal pipeline. And to do that, the course that made more sense was from an initiative called Tiny Earth. And this is starting the same way that you're student sourcing through the soil. That is, you crowdsourcing, but antibiotic or antimicrobial in general discovery. That's actually like a featured article that CIDRAP had posted from when I got trained for this course a while ago. And it also addresses the same challenge that that is posed and that you sort of heard in the previous talk as well, that we have really a dearth of antimicrobials, and it's really exacerbated in the fungal discovery. Next slide. So for Tiny Earth, the research flow is a little bit different, and the set of students that are sourced for this are different as well. These are microbiology students, where I feel most comfortable. And they're third and fourth year students as well, so they are coming in with some coursework and background before moving on to doing any kind of research work in the classroom. The flow is a little different in that students culture from soil. They then screen colonies to inhibit what are called escape pathogens. I'll define that in a bit. They isolate their best producers for antimicrobials, and it's taken into one of two directions. In fact, this is a great collaboration with the chemistry and biochemistry departments, where the students biochemically and molecularly characterize their findings, and they are learning techniques that are even at potentially protein level, and they will use NMR to further characterize much of their work. And so students now are really expanding the kind of authentic research work they're doing. Next slide. So the ESCAPE is really an acronym, and it's a good example of why you can certainly student source. And for example, with microbiology, there are a lot more sections run. So any given semester of the course, I have 48 to 72 students working to find their best antimicrobial producers. And so it's an incredible amount of findings that we comb through every semester. But the challenge of working in an institution like mine is the biosafety levels or access to some of the resources that are available, as well as the population of students that are working with these. So ESCAPE refers to, instead of using a biosafety level 2 organism or higher, to find a safer relative to test against the producers we find in the soil. And with that, we've been able to adapt, and I should say adopt as well, this project with an even younger cohort of students and really expand the reach of Tiny Earth. Next slide. So here's some good examples of just how far Tiny Earth and trying to student source and expand those efforts can really come. Those students really have come in all shapes and sizes. I talked to you about the undergraduates who are part of this pipeline now. We now have high school students as well. The World Science Festival did a antibiotic awareness campaign in spring. It's now being annually done. And this is me at Bard Early College High School with high school students doing the very same project and research flow that the undergrads did, and now have their name up with the undergrads. Down here is Ward Melville High School for an unrelated project, and that was also a sort of younger set of students, though those were 10th graders. And so we are talking like 15-year-olds working with this project as well. Next slide. So in review, compared to C-phages, which was sort of the original answer to these discovery-based research courses, Tiny Earth is much more global in its reach. There are 284 US institutions you can see on this map. It's a dynamic one too, because it's a Google map. And then about 128 international institutions, especially in India, where we see a huge rise, India, and to some extent in Europe as well, where we're seeing a huge rise in antimicrobial resistance, especially for antifungal resistance and emergent strains like Candida auris. So Tiny Earth is much more a pivotal global effort that's coming out. Next slide. In addition to Tiny Earth and C-phages being adapted as discovery-based courses for antifungals, there are state efforts. So this applies, I think, to those in the audience who may be directly from where the speakers are today, that is New York. but it also refers to any sort of state funding that's available in the United States or outside of it. In New York, particularly, we have what's called a STEP program. It stands for Science Technology Entry Program. It's funded by the state to recruit students in middle school, which would be like grade 7 through up until high school, and students who are underrepresented minorities or and/or economically disadvantaged. And over 300 students already are involved in these projects. So when we're not in session during the semester, that is in the summer, these students come out to us to do those projects. There is a collegiate version for that, the C-STEP. Many STEP students continue to be C-STEP students, and over 50 colleges in New York participate for that as well. A lot of the funding still continues to come from the National Science Foundation through what's called LSAMP. This is the Louis Stokes Alliance for Minority Participation. And it's an effort to really increase the number of URMs in order to get them to complete their bachelor's degrees and retain them by getting them involved in research within the very first year, if we can. And so these are all pictures from all over the summers, all the summers we've had, even winter. And so whenever their schools are off, they come out to us. Next slide. So in the interest of time, I can't talk to you about some of the research findings that have already come about specifically with the antifungals, but I'm happy to during the Q&A. But these are examples of 2 students who went on to be clinical lab scientists and a QC microbiologist who started out with some of these discovery-based research courses I talked to you about, working then on Cryptococcus neoformans. Their particular project involved microfluidics, and this was in collaboration then with those research institutions that I introduced to you before. Next slide. That's a little GIF of the microfluidics in action, and that's okay, Victor. Thank you. You can go to the Candida auris slide. This is another outcome, and one that we're still talking about. So those who are more domestic and closer to me now are hopefully hearing about the clusters of Candida auris we're seeing in Texas and other places. And of course, the highest number continues to be in New York. So it was of interest for us to work with Candida auris. And these are 2 examples of very good students who are first-generation college students, economically disadvantaged, who, had it not been for some of those discovery-based research courses, felt comfortable in taking on a project like this later on, working and then producing incredible data on the simple model of infection I talked to you about, as well as rodent models in collaboration with nearby institutions. And so we are creating this pipeline of students, and we're feeding them to all the local institutions where some of the incredible exceptional research that's happening that you heard about in the first half of this webinar as well. Next slide. And with that sort of like listing all the student outcomes we've had, my position as this ASM undergraduate faculty research initiative fellow is to be sort of that liaison between this teaching institution and these research intensive institutions, and create this pipeline that starts from really the bottom up to amplify all these research experiences and research findings and contribution to antifungal research and development. Most of these students go on to have authorship on peer-reviewed publications as well as conferences, which is an incredible, incredibly self-fulfilling outcome for them, but especially for someone in my position. All of my student honors have been, and they've gone on to win many, many monetary awards. And it's sort of fed itself, and the students have continued to work and truly crowdsource this effort for antifungals. Next slide. So this is sort of my thank you slide. It continues to be a work in progress to recruit, successfully retain the students, and especially some of both the challenges and the benefits that come about with working with underrepresented minorities and women in science. But it's an effort that's incredibly critical at this time when we are facing this need for building up our antifungal pipeline. I just wanted to kind of bring up the pictures again of the institutions I work with. I am here today at SUNY Old Westbury, but I continue, as you saw, I continue to work closely with all the other institutions. And so I am very localized in that sense. And then I would be happy to take on any questions or continue this discussion during the Q&A. Thank you.
Thank you, Tejas, for your presentation and all your efforts in developing the next generation of scientists across all genders and minorities and underrepresented populations of people. And I'm just inviting back Dr. Del Poeta to the Q&A session as well. So just as a quick reminder to our listeners, please stay on with us. We will now start the Q&A session. As a reminder for the audience, you can continue to submit your questions. We are monitoring these as we are continuing through this session. And include the name of the speaker you're addressing. It's probably intuitive to us, but that would be helpful, and we'll do our best to respond to as many questions as possible. So we've had many questions come in throughout your presentation this morning and this afternoon. And I'm going to start with you, Maurizio. Back to the basic scientist. You presented both state-of-the-art of antifungal research and some of your own as well, focus of your acyl hydrozones. Can you give us a little bit of background of how you started to conduct research in with these particular compound and what the mechanism of action is? What's it targeting?
Yes. Do you want? Can you give me back, Victor? Can you give me back some of those slides at the end?
Okay, I will do now.
We'll go ahead and move to that.
So I started to work on this hydrozone because we were interested to find the inhibitors of the synthesis of the glucosylceramide. So we started by doing a phenotypic assay and then a biochemical assay in which we were looking for compounds that will block the synthesis of glucosylceramide initially. We later discovered that together with the blocking of the synthesis, we found an accumulation of a different compound called sphingosine. And as you can see over here, you are decreasing the synthesis of glucosylceramide, but you are increasing the synthesis of sphingosine when you are treating with this compound. And this happened in a dose-dependent manner. This was done by TLC, which is a thin layer chromatography on the left, and by mass spectrometry, which are all those graphs on the right. So what happened is that the compounds that we discover is inhibiting the synthesis of one thing and increasing the accumulation of something else. And unfortunately, that something else, which is sphingosine, is highly toxic and is blowing up, is acting as a detergent, and is blowing up the membrane of the fungus. And that's how we start working on this. And we do not know, as of today, we don't know the precise molecular mechanism how this hydrozone is doing this. We think now that it's acting through perhaps a Golgi and the Golgi ER vesicular trafficking. We see a lot of vesicles in the cell when we are treating with this compound. There is an accumulation in the ER. We think that the primary target might be in the ER, perhaps also the mitochondria. We are studying both genomic, proteomic, and metabolomic discovery, and sometimes with the genomic we found some exciting gene. We try to validate that using proteomic, and we don't. But recently we may have some specific target which explain this phenotype using and this target might be in the ER, in the endoplasmic reticulum. And it fits very well because the synthesis of glucosylceramide is mediated through a crosstalk between ER and the Golgi, so it makes sense.
As a follow-on to that, I'm wondering about, often in antifungal treatments, you're looking for potential synergy amongst agents that target different pathways within the organism. So you did present some of that, and I'm wondering your thoughts of it as a solo agent as you develop this clinically versus in synergy with other agents.
Obviously, these compounds are targeting sphingolipids, and if you are combining this compound with the current antifungal, they are highly synergistic. Some synergistic activity that we have is less than 0.5 as a fraction inhibitory index, which is highly synergistic. It basically means that particularly with amphotericin B and fluconazole, and echinocandin also, they are synergistic, particularly amphotericin B and fluconazole. And it makes sense because they are working on the membranes, both of them. So if you are messing up, and there is a very close interaction between sphingolipids and ergosterol, and therefore if you are messing up one type of ergosterol synthesis, you are also affecting sphingolipids. And sphingolipids and ergosterol are involved in the membrane organization. So by disrupting one, you are also facilitating the action of the other one. And we have shown this in vitro, and we are starting now to also test in animals the synergistic activity of fluconazole and amphotericin B together with these compounds.
Excellent. Yeah, solo therapy, unique mechanisms of action, and then synergy, very important. Tejas, I'm going to ask you a question moving towards the undergraduate research that you've been involved in, to talk a little bit about some of the challenges that you may have unexpected in involving, or perhaps expected, in involving undergraduate students in your antifungal research.
Thank you for that question, Marnie. This is sort of one of those, I would say that most of the challenges are unexpected, although now they feel expected to me, having served in the institution where I am now, is especially with the set of underrepresented minority students that I work with, and the woman, underrepresented or not, that we're working with. And I wish I had talked a little bit about the challenges of the pandemic. We kind of didn't even bring that up. A lot of these challenges have been exacerbated with the pandemic. The students who come in, some of the pre-pandemic challenges that we would face with them is that they would not want to take ownership of their projects. Most students do not come in with, they come in with very little perception of themselves as a scientist. They don't view themselves as scientists. And it is hard to remodel that thinking for them, because as it is the case with any student who comes in with very little research background, they would prefer that you just tell them what to do, as opposed to them taking autonomy and ownership of their projects. So that was the pre-pandemic challenge. This was then exacerbated by the pandemic, because we have students who often are also, as I sort of mentioned, they're on federal or some sort of aid. And so in order to even attend college or make their courses, they had found themselves being sometimes the solitary breadwinners of their family. And so everything gets put aside, including even even if you're in the middle of a discovery-based research course. And students would often try to do much of the work, even though we never stopped doing what we did. As my college, along with Stony Brook and other colleges, went remote. And so we adapted our courses for remote delivery and continue to engage students remotely and work on everything from meta-analysis to other kinds of things. So that's how we sort of address the post-pandemic challenges. But then we had students who continued to work on these while they were at work. And it really took away from that kind of authentic research experience that we were trying to get them with these courses, including teamwork and collaboration, because students were now working so much in silo while they were trying to just kind of make ends meet, sadly. And there really were ways that we tried to address some of this, including some of the state funding that I talked about, to help many of these students. But those were, and I think students who continued to be part of the courses showed true resilience, as did we in some ways, in addressing these post-pandemic challenges. And I think we're sort of coming back from it now. And so those are some of the major challenges. And I think I'm very much focused on the post-pandemic now because that's where we are. But the number of students in our discovery-based research courses has not gone down. So despite us kind of shifting and pivoting some of these courses, we've continued our cohort, or we've continued to crowdsource as we wanted to. And so that we hope to continue doing that with the population that we have.
Yeah, certainly there's been challenges. Maurizio, I'll ask you a little off topic, but I'm sure it was similar, but how was your research impacted by the pandemic?
Yeah, I mean, we are using animals, so therefore we were allowed to be open, although staggered. So our research facility never shut down. My lab never shut down because we have to take care of the animals. Although we were only allowed to be one person in the lab at the time for a certain period of time and follow strict guidelines and things like that. Therefore, it was not, it was slowed down, not hugely impacted. But people were willing to work 24/7. So there was a lot of people coming at night, which was nice because there was nobody. People coming at different time of hours. We had an impact from a student point of view, like obviously last year we had a significant decrease of the number of applications to our PhD program. This year now we are back in business, but last year there was a significant decrease. I think that with the COVID in full swing, people just decided maybe to take a year off and wait for next year. It was impacted, but not that much, I would say, at least in my experience here. Also, my lab is a little isolated compared to many different labs. I know other labs within the institution, they have to go through the clinical setting. In that case, it's different. Our lab is located in the West Campus, as Tejas know, and here is very isolated, so it was very different. So
it's important that you're able to keep conducting the research that you have. I'm going to switch to one of the questions received for you, Maurizio. You presented in your presentation around the efficacy in the cats, your in vivo data in the cats, both with and without itraconazole, I believe it was. And the question is, how do you feel that would translate into human fungal infections?
So it's very nice. So from efficacy point of view, there is no question that these drugs are highly effective and highly efficacious. Toxicity studies that show that they are very highly tolerable. Just to give an idea, the selectivity index of some new compounds that we have, D13 was one of the parent compounds. I'm not sure if I actually am positive that D13 will not go into the human trial. We have a much more potent compound than D13. A selectivity index is about 1,000, now we are running between 1,000 and 10,000-fold, meaning that in order for the compound to be toxic, you need to increase the dose over 1,000 times. In some derivatives that we have now, over 9,000 times. This will give us a huge therapeutic window. So even though this concentration might be okay for animals, concentration from human may be scale up. I know that for some compounds normally currently in development, let's say for instance, Amplix pharmaceutical, for one of the compounds they have, a phosphogenic. This compound require very high doses in a clinical trial, and you know, they are able to do so because obviously the compound is minimally toxic, if any toxicity in things. And I think we can do similar thing for our studies. So the good thing about the animals is that they give us an opportunity to test for toxicity, and definitely we don't see that even with these high doses that we are using for animals. So that's a very, very good thing. The downside is that animals do not talk. You know, they don't tell you if there is any problem, it's a neurotic problem or things like that. So that's something that needs to be studied in human. And I think how close we are with the phase 1 clinical trial for toxicity. If you talk to me, I can say that we are close. If you talk to my CEO of the company, he said we are not. So you know, I guess there are timelines that needs to be respected, and I understand that. So we will see. A few years, I think.
Definitely a process. Takes many compounds or candidates to find that lead to take you through the clinical process. When you said derivatives, do you mean you've applied synthetic chemistry to change some of the?
Yes. Yes, we have a fantastic chemist here at Stony Brook University who is working with us for, and that's another thing, you know, if you talk to an organic chemistry, they always want to make new compounds, and it's never enough for them to say, this is enough. They always have a question: what if we change this part? What if we add the hydroxyl group in the para position instead of the ortho position? Which, you know, it can be exciting and things like that. But at the end of the day, you know, we need to move on and start testing. I think in human, the best compound that we have. I think some of the new compounds that we made are extremely promising. Yes, these are synthetic compounds. So we are working with synthetic compounds. We're not working with the natural compounds. They are extremely easy to make. They are very cost effective to make, and that's a good thing. So
stay tuned as you continue to translate your science. Tejas, I want to ask you, you talked a little bit about the outcomes, whether it was Tiny Earth or the other programs you've been involved in. And I've spoken with Dr. Jo Handelsman, and she's talked about the importance of getting that applied, getting into the laboratory and getting that experience, and how that can just spawn or that passion for science and then leads to a career in science and into graduate school. What do you have any metrics yet on how many of your students then do select to go on to graduate school and pursue a degree in science?
Thanks, Marnie. That's assessment, right? We all hate doing assessment data, sometimes post. So in all candidness, I have been involved with C-phages much more recently, but Tiny Earth since about 2017, so about 4 years of data, which complicates things a little bit with undergrads because it is a 4-year degree at least. So I don't have quite as much initial data on it, but now is the time that I'm starting to see outcomes. To kind of give you like a ballpark, figure of some of these outcomes, at least the initial cohort that we had with Tiny Earth. We've had about every single student go on to a STEM field, if not a STEM degree. And looking at graduate school outcomes, I showcase sort of the 4 students to you on the slides that I was going over. I think if I had to put a ballpark, if we're talking master's or higher degree in STEM, it is about 50%, which, to put things in perspective, the initial number of STEM graduates with the bachelor's degree moving on to a postgraduate degree or on their way to a PhD, those numbers were 25% or fewer. So just with a very small, and as a research scientist, it's not a big enough study for me to make conclusions. But in many ways, from those initial numbers from back when 2012, the PCAS report came out, I find that we've doubled it in some ways. Yeah, and I think what Joe has said, and I had the pleasure of, when I got trained, initially had the pleasure of, her leading the sessions there too. The Tiny Earth broke off from what was called a Small World Initiative, which started at Yale University. And I think that program has been in the running for quite a bit. That one does have a lot more data. I think if you kind of accumulated all the institutions that it's out there now, those outcomes, they are quite high. My lab takes it in this very specialized field where I come from as a mycologist. So these students are working on this with antifungals, which sometimes limits the kind of graduate school opportunities that they want to go for as well, because they then get hyper specialized. In fact, every single one of my students who's had who went on for a master's or PhD later on, they were pre-med to begin with. So they were invested in this because they wanted something for med school applications. And that tends to be the case for a lot of STEM majors as well. But seeing that change in the schools that they want to apply for, and then they want to go on to a PhD program, I think that itself speaks a lot more than just sort of sheer numbers, that their decision to go, that even to work on a clinical project with the interest of going to medical school and then moving to want to do a PhD in it. Having now finally, going back to what Joe said, like really developing that passion, seeing yourself as a scientist, and not just someone who is kind of garnering this experience because that's what the applications call for. That qualitatively, I think, is a very nice outcome of this.
Maurizio, that's also good for you as well, correct? Because you're looking for high caliber graduate students in this space that have a strong interest.
Yes, absolutely.
You see someone that's got an interest that has research experience from a program like this or any laboratory experience with an interest in mycology.
Yes.
That would interest you.
Yes, absolutely. Yes.
Yeah. Yeah, it just shows how it can translate into the PhD, into being mentored by someone like Maurizio. Maurizio, I'm going to ask you another question from our listeners. They want to know about the monoclonal antibodies.
Yes.
Okay.
So,
they want to know about potential side effects, and they would also have this follow-on question of do the organisms ever mutate and lose the target of the monoclonal antibody, rendering them resistant?
yes. So the monoclonal antibody therapy, again, clinical trial, one of the clinical trial that has been tested with Cryptococcus was 18B7, which was a GXM antibody. Tejas knows this very well because we have used this antibody extensively, even now, produced by Arturo Quetzalcoatl at Albert Einstein, and now is one of the major or more diffuse tools that we are using against for studying Cryptococcus. And it has been tested several years ago in clinical trial in human, and there was some benefit. But then, for some reason or other, maybe this was not enough benefit to follow up. And also the problem with the monoclonal antibody that again is extremely highly specific to the peptides or to the polysaccharides or to the lipid that are produced. We also produce, for instance, a monoclonal antibody against a different lipid, glucosylceramide. Our monoclonal antibody was an IgM. There was another monoclonal antibody which was produced by a Brazilian group called MES2 or something like that. And this was an IgG against also the fungal glucosylceramide. And the peculiarity of these two monoclonal antibody against the lipid was that the fungal lipid is different than the human lipid, and therefore was specific to the fungal lipid, and they were binding against the fungal lipid and not to the human lipid. And we show that this monoclonal antibody can work as a passive immunization, you know, it can improve the infection. But to move a monoclonal antibody into a clinical trial, it's extremely difficult, I would say, more difficult than drugs. That's because, again, the cost is extremely high to produce this antibody. And the purity of the antibody needs to be very, very good. I think now in a new technology, perhaps this antibody can be revisited. But there is always the thing that the fungus may change the epitope you're targeting, and also the fact that anyhow when you're introducing antibody, a foreign antibody, you are developing an antibody against your antibody, and so that could be a problem during treatment. And so monoclonal antibody therapy, one of the greatest success, also thing is a micrograb. Micrograb was an antibody against the Candida, and it was tested very well, and it was good. But then for some reason, again, it was not developed for clinical use. So there is a promising there, but again, in my opinion, going after a specific monoclonal antibody, it will take some time. But on the other hand, now you know everything is monoclonal antibody. So you name it, several chronic disease, they're using monoclonal antibody for everything. So it is possible that this monoclonal antibody therapy may come up in the future. You know, again, this is due to the fact that the cost is dramatically decreased compared to what it used to be. And let's say if we become tolerant to that antibody, well, no, you're gonna make another one.
Mm-hmm.
I mean, if production can be easy, it's feasible. It's very, very feasible. So
still perhaps need the, you know, you still need the antifungals, and perhaps you would use them collaboratively or synergistically. Tejas,
yeah. In my opinion, a monoclonal antibody is perhaps very, very, unless you find an antigen that is highly produced in every single fungal organism, you know, and it will be specific for that specific disease. And so to produce, to justify the cost of production with the cost of revenue, you know, when you talk to a scientist, obviously a scientist is willing to also justify that, because we don't measure a cost of life. But when you're talking to a company, they want to make money. And if the product does not make enough money, then it's a problem. So
yeah, need support, grant for research and support for developing the pipeline of students. Tejas, what, you know, are you finding enough support for your programs and initiatives to train the undergraduates or interest the high school students into science to generate more of a pipeline of scientists? Is that difficult for you?
So I think
What I mean by infrastructure, but monetarily, so monetary support.
Exactly. So yes and no. Whether you're in like a teaching institution or a research institution, support for infectious disease research alone is tough, period. And then if you work with fungi or other neglected organisms, it's even harder in some ways. You're in this sort of unique niche, but that support is hard. So in terms of federal support, there are now and I think Maurizio talked about this a little bit too, there are efforts that the NIH is pointedly making towards it too. And it's also recognizing institutional differences. So the area grant, which is, you know, in our levels is the R15 for United States, that used to be for primarily teaching institution. It's not as big a federal grant as an R01, right? That's sort of the holy grail for that level of funding. But that is for an institution like where I am, where you're working mostly with undergraduates. The National Science Foundation is in many ways supportive for things that involve broader impacts, and it's easy to sell that where we work with undergraduates, incredible broader impact to students and to the community. So the NSF has built-in mechanisms for that and many more agencies and opportunities. The NIH has finally recognized that even non-research institutions are not all made the same. LIU Post, for instance, where I was right before SUNY Old Westbury, is a health professions institution. So it trains nursing students and you know, health imaging and other allied health career students. That does not mean that they should not get or are not interested in foundational research experiences. When you research kind of provides you these problem-solving skills that are universal in any field, essentially. And so that's now been recognized. So there's the area grant has been split into institutions that are for health professions, like Post, and those that are for students who are biology majors and, you know, primarily undergraduates, like where I am now at SUNY Old Westbury. And so now we have two means of acquiring federal funding. And often a grant like that sets the precedent for then competitive awards like R01. So you can still apply for an R01, but then kind of getting to that first point of getting the R15. So that's where I am now, gets you to the stage where you can apply for that. My research focuses on quite a few variety of fungal organisms now, including the emergent species, right? And in many ways, I was trained by a clinician, and Maurizio knows this very well. He was actually he was on my thesis committee, so we have a history together. I was trained by a clinician, but I was just as trained in basic research as applied. And so my doctoral work was on aging, and we just talked about, you know, how our pathogen evolves, and that's a challenge constantly, especially with something like Cryptococcus. And so because there are research questions that are just as basic and applied in my lab, funding agencies like NSF make sense as well. And so I try to diversify my research, and that kind of allows me for going for those two federal agencies as well. All the support that I mentioned state-wise during my presentation and through Tiny Earth directly or CPhages, that makes a lot more sense for an institution like mine where I am teaching. So in some ways it's easier to get funding for teaching purposes than for research. So if you argue that I need this for my classroom, so if I ever need any strains from anywhere, including, you know, like ATCC or standard strains or clinical strains, they're given without cost because this is not for profit. and you're using this for a classroom experience. So there are ways in which you can really get some seed money, I would say, to get started, which is where I am now. And there's more recognition for federal funding for this level of research, and not
That's
all
good.
towards research. Yeah.
We have to be very, it sounds like you have to be very creative, but you are finding pockets of resources that can lead to then those larger grants that you can then target your research questions. We've come to the end of our Q&A session. Both Dr. Bouklas and Dr. Del Poeta, thank you so much. I'd like to thank everybody that listened as well for their questions and comments, and thank our speakers. And I'm going to hand it back to Victor.
Thank you. Thank you very much.
Thank you, Marnie, for moderating today. And of course, thank you to Maurizio and Tejas for your presentations. So we'll soon be announcing our next webinars. Please keep an eye out on the Revive website, where, as I mentioned before, you can also watch the recordings of our previous webinars, read our antimicrobial viewpoint articles, and also access our openly accessible resources. And make sure to keep an eye out for our emails, where you'll be receiving the links for the next webinar. So this will be all from our side. I would like to thank everybody for joining today and for contributing to this discussion. I hope you found this webinar interesting and useful, and that you will join us again in the future. Thank you and goodbye.
Bye-bye.
Thank you. Thank you so much,
Thank you.
Victor.